Soil health. Soil health is defined as the continued capacity of soil to function as a vital living ecosystem that sustains plants, animals, and humans. It recognizes that soil is not just an inert growing medium, but a complex, dynamic system teeming with microorganisms and organic matter that work together to perform essential ecosystem functions. So right there, this is exactly what we're all here for, 7+ for. Compaction on the left-hand side, and you can see the soil on the right-hand side. And Darren, Rich, those were, sorry.
What's that?
So those two samples, that was at Wilkie, they were basically 100 feet apart, weren't they, on those two separate fields?
Wilkie?
Or, not Wilkie, down south.
South Regina.
Yes.
Rouleau.
Rouleau, that's
Yeah.
it. So you can see there's a stark difference between the compaction on the left-hand side and the soil on the right-hand side. So the characteristics of a compacted soil. So the compacted soil is going to have a higher bulk density. It's not going to have as much pore space. It's going to have poor infiltration. So the water hits compacted soil and it runs off, or it ponds, or it moves to a low area, and there's a risk of salinity when it hits the low spots. It's slow drying in the spring. There's obviously restricted root growth, poor aeration, so low oxygen levels. So compaction pushes out air, and low oxygen means low microbial activity, reduced nutrient cycling, higher denitrification losses in wet years, and poor nodulation of pulses. Higher soil temperature fluctuations, so due to pore structure and lower organic matter, compaction leads to slower warming in spring, faster overheating during drought, and it stresses the crop. Reduced microbial and fungal activity, compaction suppresses microbes due to low oxygen, limited pore connectivity, fewer fresh exudates from struggling roots. So fewer exudates means a reduction in mycorrhizal colonization, nutrient mineralization, breakdown of residue, and glomalin production. So glomalin is essential for creating aggregates and having that nice soil structure. So it's also a vector, an environment for soil-borne diseases. So wet anaerobic conditions favor diseases like rhizotonia, pythium, and fusarium. Key characteristics of healthy soil, again, biological diversity. Healthy soil contains a diverse community of microorganisms, including bacteria, fungi, and other soil organisms that contribute to nutrient cycling and disease suppression. Physical structure. Good soil structure allows for proper water infiltration, root penetration, and aeration, which are all vital for plant growth. Chemical composition. Healthy soil has balanced nutrient levels, including essential elements like carbon, nitrogen, and phosphorus, which are crucial for plant growth. So how does soil health differ from soil fertility? So soil fertility is a soil's ability to provide essential nutrients and a suitable environment for healthy plant growth. It involves the right balance of minerals, organic matter, air, and water, along with the activity of beneficial microorganisms. A fertile soil maximizes crop yield and supports plant resilience, while an infertile soil can limit growth and make plants more susceptible to stress and disease. And I would suggest that a compacted soil is not very fertile. Key components of soil fertility: nutrients. Fertile soil contains essential mineral nutrients, including the macronutrients like nitrogen, phosphorus, potassium, and micronutrients such as iron and zinc. Organic matter. Decayed and animal material, humus, improves soil structure, water retention, and provides a food source for microorganisms. Microorganisms. A healthy soil is a living ecosystem supporting bacteria, fungi, worms, and other life that help cycle nutrients and improve soil health. Soil pH, the acidity or the alkalinity of the soil, measured by pH, significantly affects how available nutrients are to plants. Why soil health is important in agriculture? Soil health is crucial in agricultural soils because it is the primary determinant of crop productivity and agricultural sustainability. Healthy soils provide essential ecosystem services that support robust plant growth, enhance farm profitability, and increase the resilience of farming systems to environmental challenges. Optimal crop yield and quality. Healthy soil provides the necessary nutrients, water, and physical support for plants to flourish. It fosters strong root growth, which leads to higher crop yields and more nutrient-dense, higher quality produce. Efficient nutrient cycling. A diverse community of soil microorganisms breaks down organic matter, releasing essential nutrients in forms that plants can readily absorb. This efficient natural nutrient cycling reduces the need for synthetic fertilizers, lowering input costs for farmers and minimizing environmental pollution from nutrient runoff. Improved water management. Healthy soil structure maintained by organic matter and biological activity allows water to infiltrate more easily and improves the soil's water holding capacity. This reduces surface runoff, prevents waterlogging, and helps crops endure droughts, and recharges groundwater reserves. Diverse soil microbial communities can naturally suppress plant pests and disease, reducing the reliance on chemical pesticides. This integrated pest management approach protects beneficial organisms and maintains a balanced soil ecosystem. Erosion control. Good soil structure binds soil particles together, making the land more resistant to wind and water erosion, which is a major threat to agricultural land. This preserves the fertile topsoil, essential for long-term productivity. Climate change mitigation. Agricultural soils can act as significant carbon sinks. Healthy soils with high organic carbon content sequester atmospheric CO2, helping to mitigate greenhouse gas emissions and increase the farming system's resilience to extreme weather events. Long-term economic viability. By reducing input costs, fertilizers, pesticides, water, improving yields, and ensuring the long-term fertility of the land, soil health management directly enhances farm profitability and the economic sustainability of agricultural communities. Water holding capacity. So a well-structured soil can hold significantly more plant available water after a rain event compared to a compacted soil. The difference in water retention is primarily due to the varying amounts and sizes of pore spaces, porosity within the soil. Pore space. Well-structured soil has an extensive network of interconnected macropores and micropores. Compacted soils, the total pore volume is reduced and large pores are crushed into smaller micropores. Infiltration. A well-structured soil allows water to infiltrate much easier and faster with minimal runoff. Compacted soils have a low infiltration rate, leading to increased surface runoff and potential ponding. Water retention. Well-structured soil retains a good supply of water in medium pores, so plant available water, while allowing excess water to drain by gravity. Compacted soils will hold water more tightly in its limited small pores, making much of it unavailable for plant roots to absorb. Compacted soil is the root of all evil. A compacted soil is characterized by increased bulk density, reduced total pore space, and a lack of large interconnected pores, macropores, and high mechanical strength. These physical changes lead to a host of detrimental effects on water movement, air exchange, and root growth. So I've sort of been through this in the other sides: increased bulk density, reduced porosity. But the bottom one provides an environment in which soil-borne diseases thrive. Diseases that thrive in compacted soils. Root rot complexes, so Fusarium, Rhizoctonia, and Pythium. Why they thrive? anaerobic microsites, plus stress roots, plus shallow rooting from compaction. Rhizoctonia root rot, crops affected, canola series of pulses, compaction effect, hard pans redirect roots sideways, and I'm sure we've all seen that while we're doing crop checks. Root pruning, and they're highly susceptible to compacted soils. Pythium, so there's several different species of Pythium root rot, thrives in cold, compacted, waterlogged seedbeds. It's a major issue in early seeded cereals and peas and lentils. And phenomyces. Compacted prairie clay loams hold water. That's a perfect environment for phenomyces. This is one of the worst diseases in peas and lentils. Phytothoria root rot. Why compaction helps the prevalence of phytothoria? Saturated soil reduces oxygen and allows the zoospores to swim and infect the roots. Clubroot in canola. Compaction equals high moisture, faster spore germination, and infection. Clubroot severity skyrockets in compacted headlands, ruts, and high traffic zones. Take-all disease in wheat. Take-all is worse in dense soils with poor drainage, low biological activity. Compaction decreases microbial competitors that suppress take-all. Common root rot thrives when plants are stressed by poor rooting, low aeration, reduced nutrient uptake due to compaction. So I think the common theme here for disease is that compacted soils provide an environment where diseases thrive. Blackleg in canola. So this is an indirect relationship. The severity increases when plants are stressed, compaction limits root development, nutrient uptake is reduced, and infected residues stay wetter longer in heavy dense soils. While compaction does not cause blackleg, it increases plant susceptibility and disease progression. Sclerotinia. So this is an indirect increase. It's not directly caused by compaction, but compacted soils. There's a dense canopy. So you've got compacted soils, a dense canopy, higher humidity, poor drainage. Prolonged soil moisture, faster apothecia, so those are just spores. You get faster apothecia development. Anthracnose in lentils, indirect. Compaction, water films, increased splash dispersal. Also stressed plants are easier to infect. Reticulum stripe in canola. While VLS spreads regardless of compaction, compaction weakens plants and often results in greater symptom expression. So how do beneficial microbes prevent disease? Competitive exclusion, so space plus food monopoly. So beneficial microbes occupy infection sites on roots, rhizosphere surfaces, root tips, pore spaces. This blocks pathogens such as Rhizoctonia, Fusarium, and Pythium. How it prevents disease. Pathogens simply cannot attach, colonize, or germinate when the rhizosphere is already filled with beneficial bacteria, fungi, and yeast. Healthy soils put pathogens out of business. Antibiosis. Beneficial microbes release natural antibiotics. Many soil microbes produce antifungal and antibacterial compounds, including, I'm going to butcher some of these, phenazines, iturins, surfactants, hydrogen cyanide, genotoxins, and lipopeptides. So those molecules are produced by Pseudomonas, Bacillus, Trichoderma, and Streptomyces. These compounds directly kill or suppress pathogens like Fusarium, Pythium, Rhizoctonia, and take-all. This is one of the most powerful natural disease prevention systems in soil. Parasitism. Good fungi attack the bad fungi. Some microbes attack pathogens directly by puncturing, digesting, or parasitizing their hyphae or spores. Main biocontrol fungi in soils are Trichoderma, Colonostaches, and Coronietherium. These organisms physically eat pathogens. Mycorrhizal fungi build tougher roots. So arbuscular mycorrhizal fungi increase the root surface area, increase nutrient uptake, strengthen cell walls, improve drought tolerance. Stronger nutrient-filled roots are much harder for pathogens to infect. Mycorrhizal colonization specifically reduces Fusarium wilton root rot, Pythium, Rhizoctonia, and Aphanomyces severity and pulses via strong roots in microbial competition. Microbes improve aeration, so they're structure builders. Many microbes create aggregates, and they do this by secreting glomalin, and some microbes excrete exopolysaccharides. So these create stable pore networks. These increase porosity and oxygen, which is lethal to anaerobic loving pathogens like Pythium, Phytophthora, Aphanomyces, and clubroot. The clubroot zoospores hate oxygen. Healthy microbes literally change the physical environment so diseases cannot operate. Induce systemic resistance, and I just found out about this on the weekend. Some beneficial microbes act like a vaccine, priming plant immunity. Top ISR stimulators are Bacillus, Pseudomonas, and Trichoderma species. They trigger phytoalexin production, lignification, and stronger epidermal cells. Roots gain faster detection and stronger response to pathogens. ISR reduces Fusarium, Rhizoctonia, Pythium, Take-all, clubroot, blackleg, and reticulum stripe. Nutrient cycling microbes make plants more resilient. Microbes that cycle NPKs, zinc, boron, iron, etc., improve nutrient density, vigor, root architecture, exudate production. Strong, well-fed plants root deeper, resist infection, and wall off pathogens faster. Compaction plus poor aeration collapses these nutrient cycling pathways and diseases explode. So putting it all together, microbiology prevents disease by taking up space so pathogens can't. So in the rhizosphere, that's that 1 to 3 millimeters around the root, is completely taken up by good biology. They produce natural antibiotics, they parasitize and destroy pathogens, they strengthen roots and enhance immunity, they improve soil structure and oxygen, and they train plant immune systems. They increase crop vigor and resilience. This is why well-structured soils with high microbial activity have drastically lower root disease pressure, and why compacted soils collapse into Fusarium, Rhizoctonia, Pythium, Aphanomyces, Take-all, and clubroot hotspots. So Crop 8C Plus contains 4 different strains of arbuscular mycorrhizal fungi. Glomus intradices, Glomus mosae, Glomus agritium, Glomus etanacum, I butchered those, along with Ascophyllum nodosum, that's kelp extract, and ammonium lignin sulfate. All of these strains occur naturally in agricultural soils. So the Glomus species increase nutrient uptake, especially phosphorus, zinc, copper, calcium, and ammonium nitrate indirectly. Its hyphae explore 10 to 100 times more soil volume than roots alone. They enhance water uptake. AMF hyphae penetrate micropores that roots can access, improving drought resilience, water use efficiency, and recovery from moisture stress. They improve soil structure, so glomalin produced by the Glomus species, binds soil particles into aggregates, improving porosity, aeration, drainage, and resistance to compaction. They reduce fertilizer requirements. Because of improved nutrient use efficiency, colonized plants often need less P and sometimes less N, but this is up to the producers agronomists to alter fertilizer rates based on soil tests. They improve plant stress tolerance. They help plants withstand salinity, drought, temperature extremes, metal toxicity. AMF also prime the plant's immune system, that ISR I was talking about earlier, induce systemic resistance. They suppress soil-borne disease, so AMF colonization can reduce Fusarium, Rhizoctonia, Pythium, Phytophthora. The mechanism by which they do this are competitive exclusion, improved root strength, and mycorrhiza-induced resistance pathways. So the other glomus species that are in the crop 8C+, they all perform similar functions to glomus, inter-radices, and they're all mycorrhizal fungi. So how soil microbes influence plant growth and hormone production? So microbes produce plant hormones directly. So many rhizosphere bacteria and fungi produce the same hormones that plants make, but often in larger quantities or at key times. Auxins are a major class of plant hormones that control root development, cell elongation, and overall plant architecture. So those are produced by Pseudomonas, Bacillus, Azospirillum, Rhizomia, and Trichoderma species. Auxins, so indole 3-acetic acid. The effects on plants are increased root branching, root hair density, and overall root surface area. It stimulates lateral root initiation. They enhance nutrient absorption efficiency. And while this matters, more roots equals more NPK, calcium, magnesium, zinc, boron uptake, plus higher yield potential. Cytokinins are a major class of plant hormone that control cell division, leaf longevity, and nutrient redistribution. If auxins build roots, cytokinins build cells and keep plants young and productive. They are essential for yield, grain fill, and strong vegetative growth. So those are produced by Bacillus, Streptomyces, AMF, and some yeast. So what cytokinins do? They stimulate cell division, they delay senescence, they improve the source sink transport, so movement of nutrients within the plant, they increase bud break and branching, and they enhance photosynthesis. Microbes trigger plants to produce more of their own hormones. So many microbes activate plant hormone biosynthesis genes. This happens through root microbe signaling, especially via exudates. So basically when those microbes are interacting, the fungi and the microbes are interacting with the roots, it turns on those genes and it gets those plants producing more of their own hormones like the auxin and the cytokinins, right? So for example, the glomus species, increased root production of auxin and strigolactones. They trigger the plant to grow more fine roots, build more P transporters, and they increase photosynthetic flow to the roots. So those are the exudates. AMF upregulate genes linked to root branching and nutrient uptake. Crop 8 rebound, 71305. So it contains boron, zinc, copper, moly, manganese, iron, all in the EDTA form except boron. It comes in a different way. And it also contains peptides and amino acids. So the use of rebound or foliar fertilizers support hormone production and stress tolerance. So foliar feeds containing peptides, amino acids, and micronutrients stimulate auxins, which are essential for rooting, cytokinins, cell division, gibberellins, growth, stress reduction pathways. This improves resilience to heat, drought, and herbicide entry. If you listen to Darren's talk, he's always talking about putting in half a liter of rebound in with your herbicides just to reduce the kick that it gives to what you're spraying it on. Foliar fertilizers boost photosynthesis and energy productions. So foliar nutrients, especially magnesium, iron, manganese, and N, directly influence chlorophyll and enzyme pathways. You'll immediately see greener leaves, stronger growth, and faster recovery. What peptides and amino acids do inside a crop plant? So they improve nutrient translocation throughout the plant. They increase foliar absorption through chelation of microbes. So they act as a chelator, just like the EDTA. They reduce plant stress, and they enhance enzyme activation and metabolism. They support root development. They improve nitrogen use efficiency because you don't have to drag nitrate up into the plant to create amino acids. They increase chlorophyll production, and they aid in the transport of sugars. Phosphorus drives any production. So if you look at the nutrient analysis on rebound, it's 713, right? 13 is the phos, there's lots of phosphate. P, phosphate, is the backbone of ATP. the energy currency of the plant. More ATP equals faster growth, better recovery from herbicide stress, more efficient root development, and stronger reproductive stages. Timing is everything with rebound. If you want to increase the number of rows in your head, spray at the 3 to 5 leaf stage, and spraying after the stage improves quality and bushel weight. Thank you. I got through that in 32 minutes. I had a whole bunch of... Any questions?
Right now?
Okay.
Just more
What?
fun than income tax, Kevin?
What?
More fun than income tax?
Oh yeah, every day.
Hey, Kevin,
Yeah.
did you send that slide to us by email? Like printed out and all that?
Those, yeah. So I have a whole bunch of, so I was a real keener on the weekend. I have about 60 pages of other information here that I didn't go through while I was going through the slide presentation. I've got them all saved. I can email them to you, and I can email that
Slide presentation.
presentation. So
I asked Darren, I said, can I chat just a little bit after he's done?
I thought you were going to tell me I did a hell of a job.
You did, because you're giving me an excellent example of different tools in our tool belt. Like the last company I was in, we didn't have an individual like this. It lacked hardcore. So, Darren, if you wouldn't mind handing out the exam for what he was just talking about. Just kidding. We won't have an exam because you're not going to remember it. That's just normal. You're not going to remember it. But here's the key takeaway. We've got somebody who does. So, this is an awesome tool. He's a tool. Right, buddy?
I prefer Legend.
Legend? Okay.
Trevor, I need a
Legendary
Legend.
tool.
There you go.
This is times you will run across customers that will have a mind like what he is. You don't have to impress them with your mind. Going, you know what? You need to talk to this guy, and then you give him his number. Because this is a tool in your belt.
At 11 o'clock at night, right?
Yeah.
Well, preferably 11 midnight, something like that. But yeah, no, awesome. Like, sweet. There's a lot of information here, and well, it's nice. I don't have to learn this stuff because you've already got one. And there's not just here. There's a few other ones here.
But it's just that beneficial microbiology in the soil drives everything. Like when you listen to me talk to guys at trade shows, I'm always saying, you know, you got to treat that quarter like an ecosystem. And what's the base of every ecosystem on the planet? It's the microbiology. And it's the microbiology that drives everything. When the microbiology is out of sync or not working properly, like with compacted soils, you create an environment where disease can take over. You don't get the water infiltration. You don't get the microbes producing those plant hormones. I mean, just think about that for a second.
There's a little bit of passion here.
Like, right?
That's all I wanted to say. I'm very impressed. Thank you, Kevin. That was awesome.